Hello friends, how are you doing? And I hope you're doing good and absolutely fine. Okay, so today we are going to discuss the questions, that is the NEET pattern exercise on the chapter the plant growth and development. Let us start discussing the questions. Which one of the following hormone or growth regulators is known as the stress hormone, right? So you all know that the growth regulator can be classified into the growth promoter and the growth inhibitor, right? The growth promoter and the growth inhibitor. Right. So there are total, these growth regulators are actually, these are the phytohormones. These are the phytohormones which will regulate the growth. They will either increase or decrease the growth. Okay, so these are the phytohormones. Okay, these are the phytohormones. Right. So these are the phytohormones which are present in the growth regulator, and now which of the growth regulator is stress hormone? Now abscisic acid is always used as stress hormone, that is, it always becomes functional whenever the plant is in stress. Okay. So the phytohormone which becomes functional, the phytohormone which becomes functional during stress time. Okay, so the phytohormone which becomes functional during stress time or during stress conditions is abscisic acid. Right. For example, conditions like low temperature, conditions like reduced water or soil salinity. Okay, like in conditions like reduced water, soil salinity, right? Reduced water, soil salinity. And even low temperature conditions. So in all these conditions, when the plant is in stress, then what happens? This phytohormone, the abscisic acid, comes into play and plays a very, very important role. Okay, so the answer for this will be the abscisic acid. Let's move on to the next question. During seed germination, stored food is mobilized by. Now, right. So during the seed germination, the stored food is mobilized by what? It will be mobilized by gibberellin, right? The answer will be gibberellin. Now what will we see that the stored food which is in the aileron layer, right? Stored food in the aileron layer in the aleurone layer, right, will be mobilized by the hormone gibberellin, will be mobilized, by the hormone gibberellin, right. So the stored food in the aleurone layer is mobilized by the hormone gibberellin, and it will be acted upon by enzymes, and acted upon by enzymes like lipases, okay, like lipases, amylases and all, amylases, so that it can be utilized, the digestion, the breaking down actually of these into simpler substances will take place, So the stored food in the aleurone layer will be mobilized by the hormone gibberellin and then acted upon by the enzymes like lipases, amylases, okay. So during seed germination, the gibberellin plays a very important role in the mobilization of the food. Coming to the next question. The phototropic curvature is the result of uneven distribution of which of the hormone, right. Now we have done this in detail, you remember the canary grass, we talked about the two, father and the son, right, Charles Darwin and Francis Darwin, right. Charles Darwin and Francis Darwin, right. Charles Darwin and Francis Darwin, they conducted experiments on canary grass, conducted experiments on canary grass, okay. So both Charles Darwin and Francis Darwin, they conducted experiments on canary grass, and then they found out, okay, what they did, they took canary grass, and what they found out, that if this is a plant, now they exposed the plant to sunlight from one aspect, and they found that the tip of the plant was moving towards that exposed sunlight. That means there is some chemical, there is something in that in the top part of the plant, or that part of the plant which is exposed to light, which is moving in the direction of light, right. So they conducted experiments on canary grass and exposed this plant to sunlight on its one aspect, right. So and expose this plant to sunlight on its one aspect. The plant started moving towards that sunlight, enunciating that there is the movement of that plant part towards that light. There is some chemical doing that, helping that movement, and that was called as the phototropic movement. Absolutely wrong. So and expose this plant to sunlight on its one aspect, and the plant body, right, and the plant body moved towards sunlight, and the plant body moved or bended towards sunlight, right, moved or bended towards sunlight, showing the phototropic movement. I mean, this is one of the gestures, when you're moving, bend it towards sunlight, just thus exhibiting the phototropic movement. Okay, I forgot we are doing questions and I thought we are doing theory. Okay, phototropic movements. So that plant, that part which was more in the the apical part, that was the auxin. That was the auxin which was helping in this movement, right? So this movement was helped by auxin. Okay, this movement was helped by auxin which was there in the apical, which is more predominated in the apical region, So the phototropic curvature is a result of uneven distribution of auxin, which is more concentrated in the apical root stems, apical shoot stem, the apical meristem, we can call it, apical part of the plant. Okay, and this is one of the reasons why why it has a control over the axillary buds and does not allow the lateral growth of the plant, because of this auxin which is present at the tip and is necessary for the apical, the lengthening of the plant and all that, you can say. So the correct answer for this will be auxin, right? Okay, let us move on to the next question. Senescence or aging as an active developmental cellular process. It is an organized deterioration process, okay, in the growth and functioning of a flowering plant is indicated in, right. They say, it's senescence or aging. Senescence or aging, some call it as a programmed cell death, right? Programmed cell death they call it as, or as I told you, deterioration, organized deterioration is what is called as the senescence. Yes, senescence or aging, also called as programmed cell death, right? Programmed. This is the fourth question. Programmed cell death, where organized deterioration, okay, where organized deterioration of life processes is happening, takes place. Right? So also called as the programmed cell death, where organized deterioration of life processes takes place. And what will we see here? That yes, leaf abscission is one of the aspects, or one of the constituents of the senescence or aging, right? Leaf senescence. Now the leaf abscission, the falling of the leaves, it doesn't mean that the abscission or senescence, abscission is a part of, is one of the signs okay, signs which we can see, which we can observe from the plant of senescence or aging. So it does not mean abscission senescence will take place at only one place, or I mean senescence will take place in only one aspect of the plant. The whole of the plant can be involved in aging, a part of the plant can be involved in aging. For example, if senescence is taking place, only say in a leaf, then what will happen, there will be the yellowing of leaves and all that, and then the leaf will lose its the natural color. Yes, the turgid it will lose, it will shrink, and then it will fall off. That is abscission. Abscission is nothing but the fall of the leaf. So in that case we say that the senescence was localized. But what if the senescence is not localized and restricted to the entire plant, to the involving the entire plant, then whole of the plant will show the senescence or the aging, right. So now we move on to the next question. Right. That is, how does pruning, yes, how does pruning help in making the hedge dense? Right. So basically what is happening in pruning, we are cutting the apical meristem, we are cutting that, and what we are doing, we are helping in the lateral growth of the plant body, yes, and this makes the hedge denser. In pruning, the apical dominance, right, yes, this is a very important thing to follow, these things are all very interesting. So in pruning, the apical dominance is shut off, right, is shut off. So that the growth of the lateral or the axillary bud, so that the growth of the axillary bud will allow for the lateral growth, so that the growth of the axillary buds will allow for the lateral growth, right. So in pruning what we see, the apical dominance is shut off, and how is it shut off? By cutting the apical part of the shoot, okay, by cutting or removing the apical part of shoot. So in pruning, the apical dominance is shut off, so that the growth of the axillary buds will allow for the lateral growth, and when there will be lateral growth, that will result in the dense hedges, right, that will result in the dense hedges. And so what can be the option for this? It frees the axillary bud from the apical dominance. Yes, absolutely, this is the correct answer for this question. It frees the apical, it frees the axillary bud from the apical dominance, right. Let us now talk about the next question. Treatment of seed at low temperature under moist conditions to break its dormancy is called as vernalization and chelation. These are treatment, okay, these are the treatment of at low temperatures, much low temperature, basically to induce more of flowering and dormancy content, okay, the flowering, to induce flowering. But what we see in stratification, right, in the stratification, in the stratification what we see that there is treatment right, there is treatment of seed at low temperature, right, 5 to 10 degrees centigrade, 5 to 10 degrees centigrade under the moist conditions, under the moist conditions to break dormancy. Right. So there is treatment of seed at low temperature. There is treatment of seed at low temperature 5 to 10 degrees centigrade under the moist conditions to break the dormancy, and that is what is stratification. Okay, but when we talk about the vernalization and chelation, right, if we talk about these two, these are treatment at chill conditions. Okay, treatment of seed at low temperature. Okay, treatment of seed at low temperature to induce flowering, Okay, so treatment of seed at low temperature to induce flowering is what you know is vernalization chelation, but the true answer, that is the correct answer for this, will be the stratification. Okay. We'll talk about the next question now. Proteinaceous pigment which control the activities concerned with the light, right. So the proteinaceous pigment is the pigment which controls those activities concerned with light is the phytochrome pigment, and phytochrome pigment we already know are in two interconvertible forms, BR and PFR, that is phytochrome red and phytochrome far red, which will operate at different wavelengths. The pigment that controls activities concerned with light are the phytochrome pigments. The phytochrome pigments. The pigment that controls activities concerned with light are the phytochrome pigments. Now the phytochrome pigments are a further two type, as I told you, PR and PFR, where P stands for the phytochrome and R and FR stands for the wavelength. So PR stands for the phytochrome red, and PFR stands for phytochrome far red, phytochrome far red, and both these two will be interconvertible. That is phytochrome red and phytochrome far red, and hence the correct answer for this will be the phytochrome, right. Let's now move on to the next question. The closing and opening of the leaves of Mimosa pudica, okay, the touch me not plant, is due to, right, this is due to the thermonastic movement, right, or we can say that the basically it is a stigmonastic movement, we can say, right. Stigmonastic movement. Now this stigmonastic movement is a movement of turgor, right. The stigmonastic movement is the movement of turgor, right. This is the nastic movement of the turgor, and what we see here in this stigmonastic movement, right, or this what happens over here, that the potassium will move in from the inner cells, okay, and this potassium, because of the movement of the potassium, the your osmolarity changes, and even the water will move, and hence what will happen, there will be loss of turgidity, and hence the leaves of the plant, okay, and hence the leaves of the plant will close in Mimosa pudica, and that is why we say the closing and opening of stomata will be seen in this condition. Which combination of gases is suitable for fruit ripening? So whenever we are talking about the fruit ripening, right, we will see that the fruit ripening is always due to the gaseous phytohormone ethylene. Fruit ripening is always contributed by the gaseous phytohormone ethylene C2H4, right. Which combination of gases suitable for fruit ripening? Now here we can see that only one option with 80% C2H4, and this combination of 80% C2H4 and 20% CO2 will help in the what? In the fruit ripening. Okay, sometimes they say even the calcium carbide and many other sources of carbide also help in fruit ripening. Okay, now we use these substances from outside, so you'll all okay you will see that most of these fruits which you take from the market, what happens, you're putting these ripeners from the outside, so they will be ripe from outside, but as you go in further, you will see that they are still raw, right. We move on to the next question. Clinostat is employed in the study of, right. So clinostat, which is also called as the clinostat, it helps in the horizontal growth of the plant without being affected by the gravity. Okay, this is an instrument. This is an instrument, right. This is an instrument which helps in the horizontal growth of the plant, growth of plant, right. So this is an instrument which helps in the horizontal growth of plant without being affected by gravity, without being affected by gravity, okay, without being affected by gravity. So what we see here, right, the clinostat is employed in the study of the growth movements. Okay, clinostat is employed in the study of the growth movements. So friends, I hope you're done with this, fine and perfect. Any doubts you can always ask me. We will, in the next session, we will talk about the next set of questions. The time we meet again, a very goodbye from this side. Thank you.